Hello and welcome, dear students. Our today's topic is genomic polymorphisms. The main objectives of this module are to know about the basic concept of genomic polymorphism, to know about the types of DNA polymorphism, to understand the applications of DNA markers, and finally to know about major techniques for DNA based molecular marker detection. Dear students, let's begin with introduction. As we know and are aware about the fact that every individual differs from the another one due to the difference in their genetic makeup. The genotype is a set of genes in the DNA which are responsible for the unique trait or characteristics to make your phenotype. Organisms may look the same but still may not have the same genotype. On the other hand, the phenotype is determined by an individual's genotype and can be expressed as genes or by the visible traits such as hair color, eye color, body shape, height, etc. In this regard, we are now going to talk about genetic polymorphisms. Genetic polymorphism is a difference in DNA sequence among individuals, groups, or populations. The human genome comprises about 6 billion nucleotides of DNA packaged into two sets of 23 chromosomes, one set inherited from each parent, that is one from male and one from female. The probability of polymorphic DNA in humans is great due to the relatively large size of human genome. Genome variability includes a wide range of variations from single base pair change, many base pairs, and repeated sequences. Sources include single nucleotide polymorphisms, that is SNPs, sequence repeats, insertions, deletions, and recombinations. For example, genetic polymorphism might give rise to blue eyes versus brown eyes or straight hair versus curly hair. Genetic polymorphisms may be the result of chance processes or may have been induced by external agents such as viruses or radiation. If a difference in DNA sequence among individuals has been shown to be associated with a disease, it will usually be called a genetic mutation. Changes in DNA sequences that have been confirmed to be caused by external agents are also generally called mutations rather than polymorphisms. Polymorphisms arise through mutations. The mutation may be due to a change from one type of nucleotide to another, an insertion or a deletion, or an arrangement of nucleotides. Once formed, a polymorphism can be inherited like an other DNA sequence, allowing its inheritance to be tracked from parent to child. Polymorphisms are also found outside of genes in the vast quantity of DNA that does not code for protein. Indeed, regions of DNA that do not code for proteins tend to have more polymorphisms. This is because a change in the DNA sequences that encode proteins may have a harmful effect on the individual who carries it. Synonymous polymorphisms are those that do not have any effect on the organism and are said to be selectively neutral, as the substitution causes no amino acid change in the protein produced. This is also called a silent mutation. A non-synonymous substitution results in an alteration of the encoded amino acid. A missense mutation changes the protein by causing a change in the codon. A nonsense mutation results in a misplaced termination codon. One half of all coding sequences, that is SNPs, result in non-synonymous codon changes. Dear students, let's now discuss the polymorphisms at DNA level. Genomic variability at DNA level can be present in many forms, including single nucleotide polymorphisms, variable number of tandem repeats, for example, mini and microsatellites, transposable elements, that's ALU repeats or ALU repeats, and structural alterations and copy number variations. It can occur in the nucleus or mitochondria. Two major sources are one, that is mutations that may result as chance processes or have been induced by external agents such as radiations, ultraviolet, etc. And the second, that is a recombination. Once formed, it can be inherited, allowing its inheritance to be tracked from parent to the child. The genomes of humans may be divided into different parts based on known functional properties. The coding and non-coding regions mostly do not code for proteins. The coding regions contain DNA sequences which determine primarily the amino acid sequence of proteins for which they code. Non-coding DNA generally containing DNA sequences with no function has not yet been discovered or possibly no function exists. Such sequences may be either single copy or exist as multiple copies called repetitive DNA. Indeed, regions of DNA that do not code for proteins tend to have more polymorphisms. Recently, there has been substantial progress in understanding genome content which centered on discovered protein coding genes which consider a functional DNA sequence, moving away for discoveries of many repeat families. And various copy number variations encompass gene copies leading to dosage imbalance that plays an important role in genome structure, evolution, and diversity. The Human Genome Project has revealed that humans have only 20 to 30,000 structural genes, that is, protein coding genes. Dear students, let's now discuss the types of DNA polymorphisms. First, the tandem repeat polymorphisms. The term tandem repeats are used to describe tandemly repeated DNA sequences, also known as simple sequence repeats or satellite DNA, which includes both microsatellite and mini satellites. Tandem repeats can involve mononucleotides, dinucleotides, trinucleotides, that is the triplets, tetranucleotides, etc. Microsatellites range from 1 to 5 to 1 to 10 base pairs in length. Thus, tandem repeats with a motif longer than the upper limit for microsatellites, that is greater than 10 base pairs in length, are generally called mini satellites. Tandem repeat polymorphisms or SSR polymorphism have also been referred to as variable number tandem repeats or repeat length polymorphisms. DNA sequences that are repeated in tandem are common throughout the genomes of a wide range of species, including humans, and are often highly conserved, suggesting that they have an important function. Most research on tandem repeats has focused on those in the human genome, such as microsatellites used as genetic markers and as repeat expansion mutations causing dominant or recessive disorders with Mendelian inheritance patterns. The term dynamic mutations has been used to describe the variation in tandem repeat length that has been found in many human genes to cause a range of repeat expansion disorders, particularly those affecting the nervous system, including Huntington's and other polyglutamate disease. It should be noted that one of the genes, FMR1, which contains a trinucleotide expansion implicated in Fragile X syndrome, shows further association of permutation repeat lengths below the disease threshold necessary for Fragile X syndrome, with more complex disorders including tremor, ataxia, Parkinsonism, neuropsychiatric symptoms, and premature ovarian failure. A much larger array of tandem repeats is present in and between genes that are not known to be involved directly in diseases of Mendelian inheritance. Tandem repeats are also referred to as simple sequence repeats, microsatellites or mini satellites. These repetitive sequences can be located in exons or introns or intergenic regions, providing opportunities for the modulation of gene expression and for that of the structure and function of RNAs and proteins. Example like codon repeats translated into amino acid runs. Dear students, let's now discuss the short tandem repeats. Short tandem repeats or STRs are a very short stretch of DNA that are repeated back to back at various locations throughout the human genome. Typically, the repeating sequence is just 2 to 3 or 4 base pairs in length, and the number of copies found back to back is variable across a wide range. Unlike the case for the DNA sequences of coding genes, there is no correct number of repeats for any specific STR in the genome. They are simply areas within the genome in which variation is normal and healthy. For any specific STR, each person will have 2 copies, one that is inherited from his or her mother at conception and the other that is inherited from his or her father. Short tandem repeats are helpful in forensic and paternity testing. Because there are a lot of natural variations in STRs, the chance of 2 people matching for the exact number of repeats on both inherited copies of the short tandem repeats is fairly small. On combining analysis of many STRs across the genome, the probability of 2 people matching by random chances was found to be extremely low. The use of Y chromosome short tandem repeat loci has become increasingly common in forensic science, partly because of the loci being recombination free during meiosis and paternally inherited. The analysis of Y chromosome short tandem repeats in paternally testing of potential paternal relationships is very important, especially in cases in which a potential father is not available. In sexual assault cases, Y chromosome short tandem repeats are used to identify the male offender. Prince et al. In 1997 reported that the male DNA was detectable even in the ratio of 1:2000, that is male to female, by Y chromosome short tandem repeat analysis, the application of which was also found in human evolution, genealogical and population. Dear students, let's now discuss the copy number polymorphism or variation. So genetic association studies generally evaluate SNPs, which are variations in single nucleotide at specific genetic locations or genomic locations between individuals of the same species. Recent studies indicate that the human genome contains another frequent type of polymorphism, that is copy number variations. A copy number variation is a variation in which a segment of DNA can be found in various copy numbers in the genome of different individuals. Copy number variation range in size from a few hundred nucleotides to several megabases. Compared with single nuclear polymorphism, CNVs or copy number variations affect a more significant fraction of the genome and arise more frequently. Hence, copy number variations significantly contribute to human evolution, genetic diversity, and an increasing number of phenotypic traits. CNVs of DNA sequences are abundant in natural populations and are functionally significant, but still need to be fully ascertained. A CNV is generated by both recombination and replication mechanisms and a de novo locus specific mutation rate, which is higher than in that in SNP. Copy number variations can cause Mendelian, sporadic, or disease effects and affect gene duplications, exon shuffling, and genome diversity and evolution. Mechanisms of change causing CNV evolution in humans through deletions and duplications of chromosomal segments were described by Hastings et al. In 2009. Dear students, let's now discuss about the single nucleotide polymorphism or SNPs. A single nucleotide polymorphism is present at a particular nucleotide site. The DNA molecule in the population often differ in the identity of the nucleotide pair that occupies the site. For example, some DNA molecules in the same population may have a TA base pair at a particular nucleotide site, whereas other DNA molecules in the same population may have a CG, that is cytosine guanine base pair at the same site. This difference constitutes an SNP. The SNP defines two alleles for which there could be three genotypes among individuals in the population: homozygous chromosomes or heterozygous chromosomes with TA in one chromosome and CG in homologous chromosome. About 3 million single nucleotide polymorphisms that are relatively common in the human population have been identified, of which about 1 million are typically used in a search for SNPs that might be associated with complex diseases such as diabetes or high blood pressure. In some instances, a variant form of a gene may confer an evolutionary advantage to the species and is eventually incorporated into the DNA for many or most members of the species. And effects of the variant form may be both beneficial and detrimental depending on the circumstances. For example, a heterozygous sickle cell mutation confers resistance to malaria, but a homozygous sickle cell mutation is usually lethal. Dear students, let's now discuss the applications of DNA markers. Disease gene risk factors with multi-factor diseases. The key goal of studying DNA polymorphisms in human genetics is to identify the chromosomal location of mutant genes associated with hereditary diseases. In the context of disorders caused by the interaction of multiple genetic and environmental factors such as heart diseases, cancer, diabetes, depression, and so forth, it is important to think of a harmful allele as a risk factor for the disease, which increases the probability of occurrence of the disease rather than as a sole causative agent. This needs to be emphasized, especially because genetic risk factors are often called disease genes. For example, the major disease gene for breast cancer in women is the gene that is BRCA1 or BRCA1. For women who carry a mutant allele of BRCA1, the lifetime risk for breast cancer is about 36%. And hence, most women with this genetic risk factor develop breast cancer. In contrast, among women who are not carriers, the lifetime risk for breast cancer is about 12%. So indeed, BRCA1 mutations are found in only 16% of affected women who have a family history of breast cancer. The importance of the genetic risk factor can be expressed quantitatively as relative risk, which equals the risk for disease in the individuals who carry the risk factor as compared with the risk in those who do not. SNPs or single nuclear polymorphisms do not cause disease. However, they can help determine the likelihood that someone will develop a particular illness. One of the genes associated with Alzheimer's disease, that is apolipoprotein E or ApoE, is a good example of how SNPs affect disease development. ApoE contains two SNPs that result in three possible alleles for this gene, that is E2, E3, and E4. Each allele differs by one DNA base, and the protein product of each gene differs by one amino acid only. So each individual inherits one maternal copy of ApoE and one paternal copy of ApoE. Research has shown that a person who inherits at least one E4 allele will have a greater chance of developing Alzheimer's disease. Apparently, the change of one amino acid in the E4 protein alters its structure and function enough to make disease development more likely. Inheriting the E2 allele, however, seems to indicate that a person is less likely to develop Alzheimer's disease. Dear students, let's discuss genetic mapping and linkage. Each DNA polymorphism serves as a genetic marker for its own location in chromosome. The importance of genetic linkage is that DNA markers that are sufficiently close to the disease gene will tend to be inherited together with the disease gene, and the closer the markers, the stronger this association. The first approach in the identification of the disease gene is to find DNA markers that are genetically linked with the disease in order to identify its chromosomal location, a procedure known as genetic mapping. Once the chromosomal position is known, other methods can be used to pinpoint the disease gene itself and to study its function. The human genome contains about 30,000 genes. If genetic linkage did not exist, then we would have to examine 30,000 DNA polymorphisms, one in each gene, in order to identify a disease gene. But the human genome has only 23 pairs of chromosomes, and because of genetic linkage and the power of genetic mapping, it actually requires only a few hundred DNA polymorphisms to identify the chromosome and approximate location of a genetic risk factor. Dear students, let's discuss the pharmacogenetics and its applications. Individual response to a drug is governed by many factors such as genetics, age, sex, environment, and disease. The influence of genetic factors on the response of a drug is known fact. Study of the influence of genetic factors on drug response and metabolism is termed as pharmacogenetics. If the knowledge of pharmacogenetics is applied during drug dosing or drug selection, one can avoid adverse reactions, predict toxicity or therapeutic failure, and thus enhance therapeutic efficiency with improvement in clinical outcomes. Polymorphism exhibited by drug metabolizing enzymes is a well-known phenomena. Currently, exploration in the field of pharmacogenetics focuses mainly on the characterization of enzymes responsible for drug biotransformation, as well as on describing the various sources of variability in enzyme activity. Pharmacogenetics attempts to identify genetic variations leading to unexpected drug effects, to clarify the underlying molecular mechanisms, to evaluate the clinical relevance, and to develop appropriate phenotyping and genotype tests. SNPs can be expressed in the phenotype of the extensive metabolizer and the poor metabolizer. Accordingly, SNPs may lead to allelic variations of a protein in which one or more of the protein functions in one population are different from those in another population. SNPs and the encoded variant peptides thus provide targets to ascertain a genetic predisposition that can affect treatment modality. For example, in a ligand-based treatment, single nucleotide polymorphisms may give rise to amino terminal extracellular domains and or other ligand binding regions of a receptor that are more or less active in ligand binding, thereby affecting subsequent protein activation. Accordingly, ligand dosage would necessarily be modified to maximize the therapeutic effects within a given population containing particular SNP alleles or haplotypes. As an alternative to genotyping, specific variant proteins containing variant amino acid sequences encoded by alternative SNP alleles could be identified. Thus, pharmacogenomic characterization of an individual permits the selection of effective compounds and effective dosage of such compounds for prophylactic or therapeutic uses based on the individual's SNP genotype, thereby enhancing and optimizing the effectiveness of the therapy. Polymorphisms in cytochrome P450, which is one of the most important drug metabolizing systems, can lead to different drug responses or toxicities. Studies on SNP variation can therefore help us to evaluate the phenotype stats of the study population and to understand more about drug metabolism. Individuals with polymorphisms in xenobiotic metabolizing enzymes such as CYP, glutathione S transferase, or N-acetyltransferase have shown that an altered susceptibility towards environmentally induced diseases such as cancer, central nervous system diseases, and asthma. Warfarin is an antiplatelet drug prescribed for the prevention of stroke and thrombotic disease. CYP2C91 metabolizes warfarin normally to CYP2C92, reduces warfarin metabolism by 30%, and CYP2C93 reduces warfarin metabolism by 90%. So depending upon the genotype, conventional dose titration with warfarin could lead to either an increased risk for bleeding events or an increase in time required to achieve therapeutic anticoagulation. In August 2007, the Food and Drug Administration regulations required that a warning label be put on warfarin, which explains the relationship between genotype and warfarin clearance. So DNA markers have several applications, such as in epidemiology and food safety science, in DNA polymorphisms as ecological indicators, in evolutionary genetics, in population studies, and in determining evolutionary relationships among species. Dear students, let's now summarize the lecture on genomic polymorphisms. Genetic polymorphism or genomic polymorphism, as defined earlier, is the inheritance of a trait controlled by a single genetic locus with two alleles, in which the least common allele has a frequency of about 1% or greater. Sources include single nucleotide polymorphisms or SNPs, sequence repeats, insertions, deletions, and recombination. Genetic polymorphisms may be the result of chance processes or may have been induced by external agents such as viruses or radiation. If a difference in DNA sequence among individuals has been shown to be associated with disease, it will usually be called a genetic mutation. Changes in DNA sequence that have been confirmed to be caused by external agents are also generally called mutations rather than polymorphisms. SNPs are the most common type of genetic variation in humans. Understanding the functions of SNPs can greatly help us understand the genetics of human phenotype variation, especially the genetic basis of complex human diseases. So DNA polymorphisms comprise three types: tandem repeat polymorphism, copy number variations, and single nucleotide polymorphisms. So detection of these polymorphisms can be carried out by different methods, including allele-specific PCR, restriction fragment length polymorphism, and microarray technique, and whole genome sequencing. Calculation of sample size is very important in studying any polymorphism using specific free softwares. Dear students, it was all about today's lecture. Hope you have understood it well. Take care. Goodbye.